All right. Well, good afternoon and welcome to AAMS Journal Club today. I'm Nora Gashi, President of Albanian American Medical Society. We are very excited to have Aseda back today, and she's our devoted AAMS member and makes us proud every day. And today's guest moderator will be Dr. Tatsuo Kawai. Thank you, Dr. Kawai. So today's journal club will be about xenotransplantation. How close are we to a human clinical trial? A scientific discussion of the paper, first clinical grade porcine kidneys xenotransplant using a human descendant model. Asida Tena is a graduate student at Harvard Medical School in the biological and biomedical science with a concentration in bleeder human biology and translational medicine. Her PhD research in the laboratory of Dr. Frederick Alt at Boston Children's Hospital focuses on understanding how the brain maintains the genomic integrity in neural progenitor cells during the brain development. She utilizes advanced high throughput genome-wide sequencing technologies, CRISPR-Cas9. By the way, I checked that term.
CRISPR-Cas9, yes.
Yeah, I'm like, how do I say that? Designs, stem cells, neural progenitor cell cultures, and computational biology. And Dr. Tatsuo Kawai, PhD, he's a professor of surgery, Harvard Medical School, and A. Benedict Kazemi Chair in Transplant Surgery of Massachusetts General Hospital. He's also the surgical director of living donor transplantation and the dialysis access program. He specializes in kidney, pancreas, and liver transplantation, as well as hemodialysis access surgery. He received MD from Nihon University School of Medicine and PhD from Tokyo Women's Medical University, Tokyo, Japan. He completed surgical residency at Tokyo Women's Medical University and clinical fellowship at Massachusetts General Hospital. His clinical and research interests are primarily focused on development of methods to induce immunological tolerance in organ cell transplantation. Most recently, Dr. Kawai is involved in preclinical research of genetically engineered porcine kidneys to develop protocols applicable for pig to human clinical trials. Thank you both for doing this today. Asida, it's your turn.
All right, let's get started. Does everyone see the screen?
Yes.
All right, great. So thank you, Nora, for the kind introduction. Good afternoon, everyone, and thank you for joining us today. I am very excited for discussing a subject that I'm very passionate about. That's xenotransplantation, since I spent 8 years at MGH researching it. Although at the time when I was working on xenotransplantation, not many people knew about it, and even it was hard to explain to my friends what xenotransplantation meant. But fast forward to today, xenotransplantation is everywhere in the news. Everyone is talking about it. So I thought it was the perfect subject to discuss for this journal club. I'm equally excited, as we mentioned before, to have Dr. Tatsuo Kawai to moderate this session. Because his insights and his comments will be very valuable as we discuss the paper that was published in AJT. So thank you again, and we will get started. So what my plan for today is to give a background on xenotransplantation. It will be around 20 minutes, maybe more, depending if I have questions. Then we'll dive into the discussion of the paper, and then I want to spend some time discussing the ethical concerns of xenotransplantation. I also want to mention that I want to make this discussion as conversational as possible. So if you have any questions during the presentation, please feel free to stop me and ask the questions. Also, at the end, I would love to hear your opinion about where you stand about xenotransplantation. You know, how do you feel about it going to the clinic? So please, I would love to hear your opinions in the end. So my interest in xenotransplantation started when I joined the laboratory of Dr. David Sachs at the Transplantation Biology Research Center at MGH. Dr. Sachs is a prominent transplant immunologist, And in his laboratory, he has led many seminal discoveries in the field of transplantation. He had two areas that he was actively pursuing in research. One was the tolerance induction to human allograft transplants through the mixed chimerism approach. And this is the protocol that Dr. Tatsuo Kawai and Ben Kozumi successfully applied to the clinic. So now they have, how many patients right now, Tatsuo? Is it more than 15 that are immunosuppressive free after receiving a kidney transplant?
Yeah, something like that number, yes.
Yeah, and most of them are like more than 10 years out from receiving the kidney transplant, and they are immunosuppressive free. So they're just living a normal life like you and I. And for those who know, a transplantation is really difficult to maintain allograft function, whether it's a kidney, a heart, or lung, because of the heavy immunosuppression that these patients receive to maintain the allograft function. So the mixed chimerism approach for tolerance induction has really revolutionized in the field of allograft transplantation. But the second area of his research was xenotransplantation, and that was the use of organs from the pig to have available as donors for human transplantation, so we could end the long organ shortage that's currently looming over the transplantation field. Now, he had created a very specific line of pigs, which are called the NGH inbred miniature swine. These animals are special in a way that they are smaller than the domestic pigs, so they do not grow to like very large sizes. And they have been inbred. So he had two pigs that he started with. So for 50 years, he's been inbreeding these animals, creating very genetically specific defined loci in the MAC for this pig, whether it's in class 1 and class 2, that he has utilized for many studies in allotransplantation, but we're also using them for xenotransplantation. And this is the pig line that I was using when I was performing xenotransplantation research. Now, I was very compelled by xenotransplantation because of the numbers that come from the organ donation statistics. You can see that right now there's more than 100,000 people that are waiting for an organ donation. There is more people on the waiting list that we have organs available for them. This is more true for the kidney. There's right now over 90,000 patients that are listed waiting for an organ transplant, but only 20,000 transplants happen a year. And according to the organ donor website, every 9 minutes, someone is added to the national transplant waiting list. And on average, 17 people die each day in the US while waiting for a transplant. Now, to make this more understanding and put it more in context, worldwide, these numbers are even larger. So there is a pressing need here to have a solution for the long waiting list for organ donation. And for a long time, it has been believed that xenotransplantation, or using pig organs to end the organ shortage, was a great modality. And that's because the pigs, they have similar size, organ, and function like the humans. Now, the miniature swine, the MGH pig, is a more attractive pig for performing this kind of transplants, although we will discuss for other companies and what they're using. And having the pig as an organ donor will have unlimited supply of organs. Now, the specialty with the pig is that it can be bred and kept in clean facilities, which can minimize infection risks. But using pigs for modern medicine is not very new. We have been using pig tissues and cells for a long time. So most of you might have heard of the heart valves that we just get from the pig. The insulin, before we got the Genentech recombinant human insulin injections, the insulin was being derived from pigs and cows. And also skin from the pig has been used for the severely burned patients. So right now, xenotransplantation is coming to a bigger picture because we want to transplant whole organs or even bone marrow in humans. But for a long time, what really hindered the progress in xenotransplantation was the immune system. That's because it was found that the pigs, they express a certain sugar on their cells, which is lost in the old world monkeys and humans. So through evolution, the pigs continue to express this enzyme on their cells, but us, the humans and the non-human primates, we lost it. That means that we, the humans and also non-human primates that we use for research, we have preformed anti-gal antibodies to these sugar epitopes. So the moment that you transplant an organ that has this gal sugar, the preformed circulating antibodies will attack it and then will initiate rejection. And that was the acute phase of rejection. And no matter what we did, no matter what drugs we use, we just could not mitigate this very robust rejection. So it became really clear to us that we needed to eliminate this sugar from the pig cells in order to have any progress in xenotransplantation. But how do you create a genetically modified animal? So how do you get a pig that doesn't have this gene expressed in their cells? Well, what came to our aid was the Dolly the sheep experiment in 1996. So if you remember, Dolly was the first animal that was genetically cloned from a somatic cell. So what I mean is that for cloning, we use the cell that is not germline derived, but is a well-differentiated cell, a somatic cell. So in this case, it was a fibroblast. So what the scientists did is they took the nucleus of a single cell from the sheep and they put that inside an enucleated oocyte from the sheep. So that means that the oocyte had the cell, its own nucleus removed, and the other nucleus coming from another pig sheep cell was placed inside. You got the IVF embryo reconstruction, and that was implanted into a surrogate recipient. And in the end, you have the birth of genetically cloned individual animals. So that was the Dolly the sheep scientific breakthrough for the whole community in the science, because we can use it to genetically modify a cell in the pig and then use the technology to have genetically modified pigs. So with this technology, we can delete genes in a pig cell, or we can insert a human gene that we want in the pig cell. So for the gal sugar, we genetically deleted the gene that expresses that sugar. And then we use the nucleus of that genetically modified cell, put it into the pig enucleated oocyte. So this own nucleus came out, replaced with the one that we want, got IVF embryos, implanted that into a surrogate. And in the end, we get a animal that is genetically modified. So if you knock out a gene, you get a knockout animal, KO, or if you insert a gene, you get a knocking pig or a KI. And that technology really brought a lot of hope to xenotransplantation, because it was for the first time the birth of the gal knockout swine. So nobody believed that the genetically modification and cloning will work in the pigs, but it did. So the first group that reported it was Reviva Corps in 2002, and then our group at MGH in, so this is Goldie, the pig that was gal knockout, that with this animal, there was a lot of successful transplants that were performed at the TBRC. But what happened, because that sugar was hindering us understanding of xenotransplantation, we found that knocking out one gene revealed more obstacles. So that means that one gene is not everything that controls xenotransplantation, because there are other genes that can make it impossible for xenotransplantation to work, because there is incompatibility of other genes between the non-human primates or humans and the pig. So some of these genes, depending on the area of the research that people were involved, were about bone marrow transplantation or islet transplantation or solid organ transplantation like the kidney and the heart, or even the lung and liver. So some of the genes I want to highlight that were important for us was the complement inhibition. The complement system really mediates strong, robust rejection. That was the macrophage inhibition. This was the reticular system. So macrophages, they just phagocytose a lot. And that was more important when we were doing xeno bone marrow transplantation, and also the coagulation and thrombosis pathways that also had to be controlled through having a human gene expressed on the pig genome. So we went back to the drawing board, and this time, having identified the genes that we need, we would insert them into the pig cell. And again, through the same technology I just mentioned, you would have the birth of the animal that you want that is transgenic for the particular gene that you want. Again, on top, you would have the knockout, because knocking out that sugar is still very important. So what I performed at my research was really geared towards xeno bone marrow transplantation. And for many years, it was observed that every time that we wanted to transplant pig bone marrow cells into baboons, those cells would be cleared from the baboon circulation within minutes. So the macrophages would just like engulf all the pig cells. So that was almost like zero survival of these cells when transplanted into the baboons. And also we got no engraftment because they were clearing out. They had no chance to engraft into the bone marrow. The problem there was the CD47 gene. So the CD47 gene is very important to mediate an inhibitory signal to the macrophages, whether they're from the baboon or the human. And the pig CD47 gene is not evolutionary conserved between the human or non-human primates. That's why it does not properly signal to the macrophages to get inhibited. So the macrophage, when it doesn't see an inhibition from the CD47 from the pig, it doesn't recognize this inhibitory signal, so it will just eat up that cell. Now, the same process also happened in our human bodies. So the CD47 is very important. Also, it's expressed on red blood cells, and aging red blood cells, they downregulate the CD47 gene. So in the spleen, where most of the old cells, red blood cells, go to die, they are cleared up by the macrophages that are in the spleen. And it's because these old red blood cells that downregulate the CD47 gene. So with that knowledge, we just really knew that we had to create a transgenic animal that would express a human CD47 gene if we wanted to have some level of success in xeno bone marrow transplantation. And that's what I did for almost a year and a half. I was genetically modifying the pig genome to express the human CD47 gene. And on March of 2012, the first human CD47 transgenic animal was born. And he was different from any other one because he was having ubiquitous expression of the CD47 gene. So this transgenic human gene was expressed in all the cells and organs of the pig. And it's almost like 10 years ago, this March 2012, and like we almost close to the March 2022. But with this animal, I performed many xeno bone marrow transplants, and we showed for the first time that having the human CD47 gene on the bone marrow cells of the pig, it really led to an increased level of pig peripheral of pig cells circulating in the baboon periphery, and also they lasted for a longer time, up to 8 days, in comparison to pig cells that were transplanted from a non-human CD47 transgenic animal. So this was very important, and CD47 gene continues to be an important transgenic for all of the modifications that have happened subsequently. But we had a limitation in how many genes we could actually put inside the pig genome. We wanted to delete many other genes, and we also wanted to insert many other genes that were important, but the tools that we had available at the time were limiting our possibilities about creating a 10 or 12 pig modified genome edits. But in 2013, the CRISPR-Cas9 actually became available. And this technology has really revolutionized the field of genome editing, not only in research, but also in human medicine. The CRISPR-Cas9 is so powerful that it can allow you to do up to 25 or even 60 genomic edits precisely in any species that you want. The CRISPR-Cas9 is what I used also in my laboratory for studying the brain cells. It is such a powerful tool that nobody has seen before. But this also would be the subject for another journal club, because I'm also so excited about this, and just science in general is just so cool, because it has also the potential to revolutionize the field of medicine for correcting many of the monogenic diseases that actually we don't have a way to correct in humans. And you can do live gene editing with the CRISPR-Cas9. Now, this powerful gene editing technology was discovered in the bacteria. This is part of the bacterial immune response. So bacteria is very smart. If it gets invaded by a virus, it can just chop out the virus DNA and insert it in its own genome. So this will be the red part. This will be viral genomic DNA in the bacterial gene. Then the bacteria has a protein called Cas9, and these act as the scissors. And also a guide RNA, this is a 20 nucleotide long RNA that acts as a guide to direct the scissors at the precise genomic location that you want to cut. So if you want to do genetic modifications, you just design a guide RNA, and then this guide RNA would guide the scissors where to go, and they would cut. And you can delete a gene or you can insert a gene. So with this powerful technology, you can do as many modifications as you want. And because it has been used now even in humans for correcting beta thalassemia and also in other basic research science, in 2020, the two women that discovered this powerful gene editing technology, Emmanuelle Carpentier and Jennifer Doudna, were awarded the Nobel Prize in 2020. I think this is one of the most well-deserved Nobel Prize that I've seen in the field of genomic editing and also in chemistry in general. But with the powerful CRISPR-Cas9 and what we can do now with pig genome editing, in 2015, for the first time, scientists from here at Harvard Medical School showed that they could CRISPR out, or they will cut out 62 sites of the porcine endogenous retrovirus, the PERV, through the CRISPR technology. So the FDA has always had the concern that the retroviruses would be a possible pool of infection in human clinical trials. So what this group did using the CRISPR, it cut out the 62 sites of where the PERV is located in the pig genome. In 2017, they reported that they had created the virus-free piglets. And then in itself created like another momentum for xenotransplantation, because they were in the news everywhere. And eGenesis, the company that Tatsuo collaborates with, was actually founded to also create these germ-free animals, but also insert or delete the genes that we want or don't want, so that we can make them available for clinical human transplantation. So with the powerful genetic editing technology that CRISPR-Cas9 made available to us, other companies such as RevivaCor and United Therapeutics have moved on creating what they called the 10 gene edited pig that is like the key to xenotransplantation. So with this powerful technology, they have created animals that are available for heart and kidney transplantation. So with the CRISPR-Cas9, they had knocked out 3 pig carbohydrate genes and also the pig growth hormone receptor, GHR. Now the growth hormone receptor was important because if you are using an outbred pig, so not the MGH inbred pig, the organs can actually overgrow. So we do not want them to overgrow when transplanted into a human. So the scientists CRISPR'd out the pig growth hormone receptor. They also added 6 human genes that are important for controlling anticoagulation. There are 2 complement inhibitor genes and also the 2 immunomodulatory genes such as the CD47 and heme oxygenase 1. So these are the genes from RevivaCor and United Therapeutics, but all the time the question is, do we need all these genetic modifications in the pig to have a successful preclinical trial or even phase 1 clinical trials when we're talking either about the heart or the kidney? So that's all. What are your thoughts about this?
Yeah, we still don't know which transgene is really necessary to insert, and definitely triple knockout pigs, just deleting alpha gal and beta for GNT2 and SHIMA is helpful, but it's still controversial whether we should remove the growth hormone receptor genes. Mm-hmm. And probably in terms of inserting genes, it seems to be important definitely important for complement inhibitory genes like a DAF or CD46, but thrombobonulinum, maybe it's important for a cardiac transplant. But we still have to do a lot of control to find out which transgenes important.
Yeah, I agree. So as a follow-up to Tatsuo's comment, Dr. Megan Sykes, who also is the director for Center for Translational Immunology at Columbia, together with Dr. David Sachs, have shown that actually the CD47 gene ubiquitous expression in the kidney actually creates a pro-inflammatory and prones the kidney to swelling. So it doesn't really help having CD47 expressed in the kidney. It's very important for xeno bone marrow, but it's not important for the kidney or potentially even for the heart. So she has her reservations about certain genes being expressed, all the genes expressed for the heart or the kidney. So in general, you would want to have every organ tailored for either knocking out or knocking in the gene that will increase the survival or control the immune rejection. And perhaps in the kidney, you don't need the CD47. I mean, there's a discussion in the end about the paper with the problems that they saw, but that could not be, due to the CD47 expressions. They were using a brain dead patient for the experiment. But again, these are problems that still require a lot more research to actually really understand which transgenes are important for what organ, so that we can not add genes that might be harmful down the road. So with that, I would like to briefly discuss now the most discussed story ever, which was the first human xeno heart transplantation that happened at the University of Maryland. But prior to that, the University of Maryland didn't just transplant the pig heart into the patient. That actually was a lot of knowledge that have acquired for almost longer than a decade in pre-clinical models of pig to non-human heart transplants. And this work was led by Dr. Mohamed Mohudin, who is the director of the cardiac xenotransplantation program at University of Maryland. And he has extensively worked with xenoheart preclinical models. In 2016, they showed that the pig hearts could remain healthy for more than 2 years, but this was just a heterotropic heart transplant. But since then, they still have done also orthotopic transplants and have shown that the heart can beat healthy and sustain life. A group in Germany in 2018 also reported that using the pig revivar-cores, genetic modified animals, they had pig hearts survive up to 9 months in non-human primates. So that's a very long time. Usually the FDA will require anytime between 6 to 9 months preclinical data to show a validation of this technology. But what the scientists also found is that it is very important to have the pig heart preserved into a solution and perfused with nutrients and hormones prior to transplantation. This is not just simply putting a heart on ice does not work for xenoheart. It needs to be really perfused and preserved in a special solution that the German group had developed. And with all that knowledge, that's how the University of Maryland performed the first xenoheart transplant on January 7th. The recipient was a 57-year-old patient who had heart failure, and he was deemed ineligible for the human heart transplant. The team had applied for a compassionate use authorization with the FDA, so that's how they gave the green light to go ahead with this experiment. But apart from using the genetically modified pig with those 10 gene edits that I just spoke before from Reviva-Core, the group also gave the patient a very strong immunosuppressant called anti-CD40, which is a co-stim blockade developed by the Kiniksa Pharmaceuticals. And for those of you who don't know what co-stimulatory blockades are, usually B cells and T cells, they work together, especially when B cells need T cell help to actually produce antibody in response to an antigen, whether it's an infection or in this time, an allo or xenograft transplant. So this interaction is very powerful, and you need the MAC and TCR first that's going to interact, but a second signal, which is the co-stimulatory signal, is important to provide that strong immunological synapse. So what the scientists did, they developed an antibody that would block the CD40 on B cells. So the interaction between the T cell and B cells will be suppressed, and you will not have these strong antibodies that would mediate rejection. So that's where the patient is at right now. As far as my understanding is, he still continues to be well. Is that true, Tatsuo? Do you know more about it?
Yeah, he's, as far as I know, he's doing great off ECMO, and
Yeah.
his ejection fraction is 65%.
Wow. That's great.
But he also developed some aortic dissection, which was surgically treated, and because of this, he developed kidney failure. So he's on dialysis.
Oh boy.
But heart function looks, okay, stable right now.
Mm-hmm. So yeah, I guess with time we will know what is happening to the patient, but at the moment it seems.
Was the dissection secondary to the transplantation or inherent to the transplantation?
That's what I don't know. I think it's just complicated after the transplant.
Okay.
Yeah, they did say that they have to make some modification because the anatomy wasn't what they expected. I mean, it was close, but they had to make some modifications to accommodate the heart in the chest. So maybe that is secondary to that. But I'm sure that there will be a full report from University of Maryland about what they have found so far. But nonetheless, this is the first time. I would never expected that the heart would be the first to be tested in a human, but it happened. And I mean, I want to emphasize that this is no way meaning that now the FDA will authorize clinical trials for xenoheart transplant, because there's a lot more research that they need to see to prove and further validate the technology. But nonetheless, it was a procedure that was really cheered for everybody that does research, because we wanted to see these tested in the clinic, because no matter how much we test this in the baboons, we'll learn and know how to better treat patients with xenohearts once they can have a large clinical trials. So these tests actually will have a lot of information that will be important to us. But prior to getting to the paper, I want to mention this news that also came last year in October, when Dr. Robert Montgomery at NYU Langone Transplant Institute showed that with the family's consent, they had attached a pig kidney that was genetically modified into a brain dead patient. But the kidney transplant was not actually done in the standard way that everybody's doing them. They just for a proof of concept, what it did was attach the kidney to the blood vessels of the patient's upper leg outside the abdomen, and it just observed whether the kidney would remain pink or even produce urine. So this was just a very preliminary observation, but we didn't really learn a lot from it, because we already know that from attaching kidneys to non-human primates, the kidney remains pink and it produces urine. But it really, brought again a momentum and people becoming aware of the advances in xenotransplantation, and possibly with every knowledge that we have gained from all the preclinical models in pig to non-human primates, perhaps is going to be a way to create a clinical trial. I think there's a question in the chat. No. Okay. So before I go to the paper, is there any question about the background?
I had a quick question, actually. What was the main reason that most groups that are studying this are using pigs instead of using a primate to work on genetic modification, since it sounds like there's quite big challenges with some of the the different genes that they're expressing.
Well, I mean, I'll give my comment and then Tatsuo can also give his, but I mean, first of all, non-human primates are, a reservoir for many viruses and infections that we cannot properly control. And once we do those transplants, for example, if we wanted to in humans, that would be an outbreak of infections that we would not know how to control. Second, I think the organ size is smaller in non-human primates compared to the pigs. So that would be a problem for patients that are, for example, 80 kgs or 100, so larger patients. So it will not be compatible, the size of the non-human primates with that of the human to sustain function very well. And third, I think they are species that actually are as endangered, so we cannot really breed them for, having them as a source for transplantation. So I think that's why the pigs have always been to be the friendlier way to go for creating genetic modifications, so we can have a pool of organs for transplantation. I mean, that's what are your thoughts of why we're not?
Pig is ethically more acceptable. Also we can make organ, tons of organ in like industrial levels, but we cannot do that in primates.
Yeah, I think that the gestation time for pigs is also shorter, so we can quickly generate the genetically modified pigs quicker. It's only 3 months, 3 weeks and 3 days. I don't know about 3 hours and 3 minutes. But these gestation times provides us a quick way for creating the animals that we want versus the non-human primates. But I think the zoonotic here is like a major concern, that we do not want to create any outbreak from potential viruses that could cross into humans in immunosuppression patient, and then there'll be ways that we will not be able to manage those infections. And pigs are generally.
I could tell you that based on my experience, when we were doing this in the late 70s and early 80s, infection was the #1 reason that people did not like using primates.
Yeah, I agree. I mean, still the thought of AIDS coming from monkeys, is very disturbing. And we don't know what other viruses that they carry that could potentially be very infectious and deleterious to humans.
No, it makes a lot of sense, and all those are really good reasons. It's just that all the issues that you brought up on the background, I was very thorough, by the way, so thank you for that. Including the multiple issues with genes needing to be knocked out, genes needing to be expressed. It's basically trying to put the Humpty Dumpty that broke up back together, piece by piece.
I mean, true,
Very challenging.
It is challenging, but I think with the CRISPR technology, we can do it so quickly right now that it has made everything very easy. So before it was very challenging with the tools that we had for genetic modification, but the CRISPR-Cas9 is so powerful, can do it so quickly, and then we can create pigs that are free of viruses, and then put in genes that we want or we don't want, take them out. But again, I think as Tatsuo mentioned, we need a little bit more preclinical research to really pin down which genes are important for what organ when we transplant them.
I echo Lawrence that definitely your information about mechanism of complement-mediated injury was very thorough. And obviously, talking about implication of cell fusion and mechanism of transfer of viruses between species, like given just in humans, the viruses that we get, including as simple as cytomegalovirus and these, were they studied on the xenotransplantation at all, or there was not an opportunity for that?
CMVs definitely have been studied and have been shown to be important to control. So we have animals now that are being born into CMV free. So even before transplanting them, we always check these animals whether they are CMV positive or not. Because if they are CMV positive, the CMV can, in an immunosuppressed, even non-human primate, can cause a lot of problems.
I mean, CMV is as simple as that, but the other thing that potentially can be BK, like things that potentially can jeopardize the kidney.
Yeah, those have not been, at least in my experience when I was there, those other viruses were not deleterious. It don't show any problem apart from CMV. That's what, have you seen anything else from your recent preclinical?
Well, it's been a part, posting endogenous retrovirus is most concerning. But now we have PERB-free pig, so that concern is absolutely eliminated. And the other virus, CMV, they will be raised those virus-free environment. So I just don't have so much concern about those.
This picture came out nicely.
Say that again, sorry.
Said, may I ask a question
Yes.
before you move on? If they can clone Dolly from a cell, where
Mhm.
can I volunteer?
We can't do that. They shut down those kind of research for doing human embryo genetic modifications.
I'm sorry.
Everybody.
I'm at the end of my life and I was looking for a reprieve, but okay.
I mean, yeah, we all would like to have a clone of our own, but unfortunately we can't do that in humans. Can do it very successfully in the sheep and the pigs now that we have shown. But yeah, we can apply the technology to humans.
So
So-
sad.
It's not a technical issue, right? It's more of an ethical issue. right?
Yeah,
They-
yeah.
They're
Oh,
already
well,
doing. yeah.
Lawrence, I was joking just in case.
We need you, Dr. Ushas. You can
Okay,
leave
sorry.
us anytime. We need to
I
clone
was
you.
joking, but
Seda, that should be our next journal club about the
Oh,
ethical issues. We
Oh,
talked
yeah,
about
yeah,
that.
sure. With the CRISPR-Cas9 is
Uh-huh.
issued a lot of problems for the human embryo genetic modification specifically because the Chinese scientists CRISPR'd out the CCR5 gene on two human embryos and then created these CCR5 free infants, which is completely outraged the whole community for trying to do such, meticulous genetic modifications in human embryos because we still do not understand everything about, the human development and also the consequences you can have from CRISPRing out the gene. And so all of the kind of research has been like shut down right now. But I mean, if done correctly, that technology is very powerful and can eliminate the monogenic diseases that are actually, inherited. And we can just potentially eliminate those inherited monogenic diseases through the CRISPR-Cas9 of the human embryos. But again, that's under tight regulation right now, and nobody's really doing even the research. But yeah, that's another very important, that's how powerful the CRISPR technology is. So that's why I said I feel like it would be worth another journal club discussing it. So, well, let's move on because I think we're running close to time on the paper. So the group here, sorry. The group, go back. So the University of Alabama group just went a step further from what the Bob Montgomery group had done. So they wanted to create a xenotransplant trial by using a human dissident model, but at least they would do the orthotopic kidney transplant right now and look for signs of rejection, and also whether there was transmission, or also assess how instrumentally ready the group is to actually have these be applicable to phase 1 human clinical trials. But we will talk about what they found and the model they're using. So anyway, for this trial, they had to get approval for both the animal facility and also the organ procurement and treatment. So that's the ICOOC approval, and from the using for xenotransplantation of the dissident, they also had an IRB approval to perform this kind of trial. So they, the overall goal here was to see whether the genetic engineering is it sufficient to prevent hyperacute rejection. So they would look grossly at the appearance of the kidneys after they were transplanted. And they developed a very new cross-matching histocompatibility assay that, for allograft transplant is widely used, but for xeno transplant has not been used. I mean, the lab, we have our own ways of determining this, but they had created specifically a cross-match assay that is done through flow cytometry to see whether the negative prospective cross-match was sufficient to prevent hyperacute rejection. So what I mean by that is that does the recipient have any preformed antibody that will actually initiate rejection? So in terms of the surgery, are there any life-threatening intraoperative complication, and would there be any derived products detectable in the human blood? And as I mentioned, a secondary goal here was also to test whether their xenotransplant program infrastructure was well established to execute all the steps required for a xenotransplant in living humans. So the study timeline here and the event summary that they had. So pre-transplant, the brain dead patient underwent a bilateral native nephrectomy, and the 1-year-old 10 genetic edit pig also underwent bilateral renal xenotransplant, xenograft procurement, and they were transplanted into the brain dead patient. So the right kidney was anastomosed in a ureter to bladder fashion, but the left kidney, the ureter was just gone through the skin as an end urostomy. So I don't know why the difference between the two anastomosis of the ureter here, but they did seem to have some complication from these two approaches of the anastomosed xenografts. And then they followed the kidney function for 3 days. And at the end of the day 3, they removed the kidneys to do further gross assessment and also histology assessment. So this is the demographics of the patient. He was brain dead because of head trauma. And he was around 57, he was 57-year-old. And the patient was declared 5 days brain dead before he was deemed eligible for this xenograft transplant trial, and was maintained on phenylephrine, the vasopressin, levothyroxine, and steroids, and had normal hemodynamics. And this is the route of management that they have for brain dead individuals prior to organ donation. So a next of kin was approached about the decedent to have his authorization to have the patient enrolled in this study. So these are the test analyses that were received prior to the xenograft transplant from the brain dead patient. Now, if you look at this, I highlighted some of the problems that I see here, but there was an elevation of pretty much everything here. So having a patient brain dead for 5 days, it just creates a very, hostile, inflammatory milieu. So it's not the ideal environment to want to test the functionality of a xenograft. I mean, the patient before had high BUN and creatinine, and also his PTINR and D-dimer were very high. His neutrophils were very high. Platelets were low, and they did say that around day 2, he started having DIC. So it was really difficult to assess how well the kidneys would function. So that's what were your thoughts about using this kind of approach to assess the xenograft?
This is kind of easy to be publicized, but it's kind of, I personally, I don't like it at all, because, transplant into the brain dead person, as I said, that told us, very inflammatory environment. And usually it's very difficult to transplant, even in allograft, put the allograft into the inflammatory environment is really bad.
Right.
Also, this is a xenograft. This makes it much more difficult. So the entire purpose of this is probably just to make sure technically feasible, but I think that can be tested other way. We don't need to use humans. So I just don't know why they have to do this.
Yeah, I also question that, but I think that in general, at least from giving them the benefit of the doubt, they wanted to see whether there was any immediate acute rejection observed here, but we already know that. So from many non-human transplants that have been done, that you won't see that because there's 10 genetic modifications that you have mitigate that acute rejection, and test whether this was feasible from that end to coordinate everything in case they can go to the FDA for a phase 1 clinical trial. But again, I don't think that this actually justifies, I mean, if FDA sees this, I don't think that they will be very, impressed with the findings, at least that we'll discuss now with the kidneys. Because the brain dead patient is not the ideal patient you want to test this. But okay, we'll go ahead. This is just their cross-matching assay that we discussed, that this is a novel assay that they mentioned. So they test prospectively and retrospectively in the brain dead patients. So what you do is you get the patient's serum, and then you mix it with peripheral blood mononuclear cells, PBMC from the donor pig, and then you look for antibodies whether you have preformed antibodies that would bind to these peripheral blood PBMCs from the pig. So they had screened many human sera in their center, and they identified negative controls, but they also identified a patient serum that had positive control. For unknown reasons, this patient was cross-reactive with these pigs donor PBMCs, but for the decedent brain dead recipient, we can see here that both before and after there was not cross-reactivity. So that means there were no preformed antibodies before and after the xenotransplant. So for their pharmacological immunosuppression regimen, they chose the conventional immunosuppression that is utilized in kidney allograft transplant, just to mimic a real world experiment for this trial. So they did B-cell and T-cell depletion, and then they had CNI calcineurin inhibitors with tacrolimus and MMF and the steroids, so the standard immunosuppression seen in kidney allograft transplants. So let's just go grossly. This is the pictures from the right kidney and the left kidney on post-op day 1. So overall, the kidneys look nice and pink. These red spots, there is blood under the capsule here on the right. They say it's due because of the biopsy. So that's why you see this blood accumulation. You don't see it on the left. They still appear grossly well on POD3, although they do mention that the kidney capsule was very fragile. And I never seen that in the xenografts performed on my center, but that's what, do you see this when you, from many genetic modifications, when you do this transplant, do they have a really thin capsule?
Yeah, pig has a very thin capsule compared with the humans. So
I mean,
it's not unusual. Yeah.
But do you see this kind of tearing in your transplants?
It's
at the time that you explant, do you see that the kidney is well intact, or does it still have signs of capsule tearing?
oh no, it's usually it's normal.
Yeah, so
Capsule tear, probably its kidney was swollen. That's why.
Yeah.
It's capsule tear.
Yeah, so at the time that, yeah, both the left and the right kidney did have signs of swelling, but that's also because the patient was in DIC and hemodynamics were very unstable. So it could be that, there was no way for these kidneys to properly function in a environment that would not sustain its proper function. And this is just grossly after explantation, how the kidneys looked. So, I mean, looking at the overall function, the right kidney did produce a lot more urine output compared to the left. But what's more concerning is that the BUN and creatinine, they actually progressively went up. So the kidney was not filtrating very well, if not at all. So this remains a big problem if you want to assess the xenograft functionality in the humans. But again, given the constraints of the modeling here, that definitely can affect the interpretation of the data here. So I don't think that this really reflects the incapability of the xenograft to actually properly filter. I think they can. It's just that where they were transplanted really affected the kidney function filtration. And from the urinalysis, they see proteinuria, it's pretty high, 2+, hematuria, and also glucosuria. So overall, the kidneys were not functioning very well, especially the left kidney. But here for both BUN and creatinine were high for both xenografts.
I think the reason the kidney didn't work is that probably immunosuppression is bad. Probably they don't understand immunity in the xenotransplantation very well. Because triple immunosuppression like tacrolimus and Cerecept and steroids, it's against the cellular rejection, right? The T cell, the T cell rejection. But the biggest problem in the xenotransplant is to suppress the innate response.
Mm-hmm.
So to suppress innate response, we have to have a costimulatory blockade like a CD40 or CD40 ligand.
Right.
Definitely that is important, but they I don't know why they didn't use it, but the triple immunosuppression for the regular allo transplant usually doesn't work at all.
Yeah, for the xenotransplant.
Right. And actually what we are seeing is once we overcome innate response, the cellular response against the xenograft is very weak. That is my impression. So the initial innate response is so intense. So we didn't know that cellular response is so weak. That's why once we overcome, then overcome innate response, maintenance immunosuppression is much less than the regular kidney transplant.
Well, that's a good
That's
point.
my impression.
Yeah, I mean, I agree. So, yeah, I don't know why they didn't use the costim blockade, but they, justifying the use of the standard immunosuppression, conventional immunosuppression in kidney allografts. But yeah, for xenografts, I agree that would be different. But I don't know, like, it really how much this was a contribution to the rejection, or that because the patient just had a very inflammatory status, so no matter what you gave as far as immunosuppression go, it just was not the right environment for these kidneys to work, because everything was just upregulated here. And just the final figure here, looking at histology, this is the patch versus silver staining. So C4D is a marker for antibody-mediated rejection. They say that throughout the right of left kidney from day 0 to the explant time, they don't see any C4D deposition, but there are some areas here that do stain positive for the C4D. But overall, it's not like you have a diffused C4D staining. And for the H&E stain here, they do say that for both A and B here, so this is day 0 pre-implantation, they see some mild to moderate tubular injury with from cold ischemia. So this is expected. But from day 1 to day 3, they actually see this diffuse glomerular congestion with swollen endothelial cells. That is consistent with thrombotic microangiopathy, so TMA. So that does remain a concern. And for the later days from day 3 to explant, they see acute tubular injury. There's some blood in here, and continues to be still evident at the time of explant, the tubular injury. So I think that overall, the kidney did have a hard time functioning well. So that's my take on it. But for the general questions that they wanted to answer, by doing this experiment. Would there be hyperacute or immediate rejection? They saw nothing. It didn't happen. Would a preclinical test of compatibility with cross-match identify hyperacute rejection? Sure, yes, they have the assay. Would a life-threatening intraoperative complication during the kidney pig xenotransplant be seen? They did not see that. Actually, both the kidneys were quite accommodating for the regular anatomy that we would do allograft transplant. Would pig-derived cells be detected in the blood? They did not detect them, not even the viruses were detected, although this is only 3 days, so I'm not sure that's long enough to make that statement. And could the pig kidney xenotransplant be safely performed under the conditions necessary for clinical trials? They say through this environment, yes, but I don't fully agree to that question. So let's just spend a few minutes quickly here on ethical concerns. So everybody has the concern about the long-term outcome of human health with pig organs, especially coming from these zoonotic viruses that might be a concern. Although, you know, with the CRISPR-Cas9 genetic editing, we can really cut out these, like we did with retroviruses, potentially CMV or other viruses that are potentially harmful to humans. Now, organ allocation and facilities to generate pathogen-free pigs. Now, this is the most important one. So there's a rush here of 3 big biotech companies that are racing to have a phase 1 clinical trials, and United Therapeutics, which is ahead of the game here with that 10 genetic modified pigs, they actually have a facility right now that they obtained the license to create the state-of-the-art facilities for creating these pathogen-free pigs, of course, that are also genetically modified. The second one is the Genesis, that's the biotech company that Tatsuo is working with. And the third one is Makana Therapeutics that was acquired by Recombinetics. This is from a group at the University of Miami from Joseph Tactor. So he's another transplant immunologist, xenotransplants. Public consent. Now, this is a big one because not everyone can actually welcome the xenotransplantation. Most people are still, they are having aversion from having animal organs being transplanted into humans. There are the Muslim groups, for example, they would be opposed to receiving a pig kidney transplant for religious activities that they have. Now, the distribution of the medical expenses is also a concern because who will pay for this? Is the health insurance going to cover all the expenses? So these are big questions, and that a lot of regulators at the FDA and also the hospitals that have these established, transplant centers are working to try and address them, because these ethical questions and concerns will also be addressed before the FDA will actually give the green light to start the phase 1 clinical trials in humans. But with everything that we've seen so far, within an hour, you can potentially cover everything about xenotransplantation. But the general question is, like, have the recent studies really given us enough strong evidence to initiate a pig to human phase 1 clinical trial? So my answer is, I'm on the reserve side, not yet. There's still work that needs to be done by using the pig to non-human primate models until we have very clear understanding of what these genetic modifications are doing and having the right combination of immunosuppression so that we can maximize success once we are ready to translate this into humans. So that's what are your thoughts?
I think getting closer is much better now. We have 2 monkeys surviving without rejection or TMA for over 430 days now. So then a couple of other monkeys also following. So eventually, the problem is that the result is not consistent yet. Right. Some monkeys, develop TMA within 100 days. We're doing the same thing, but also using same genetic modification, but still having some rejection, and some monkeys doing great without any rejection. The other thing is, the other thing is, if the human is supposed to be better than monkeys, because monkey has still antibody against the TKO pig organs, but human doesn't have it. So monkey is kind of more difficult than humans. So if we're successful in monkeys, we can go ahead. But the other hurdle is that we have to, I think we have to use anti-CD40 ligand and the anti-CD154 antibody. But CD154 is not clinically available right now because of thrombogenic complications. So right now we also working on the anti-CD154 FC modified non-thrombogenic anti-CD154. So we're gonna our plan is to start clinical trial of that antibody this year, sometime this year. Then once it's done, then the antibody is available in time for xenotransplant. So that's then great. We can go ahead probably next year, hopefully, or following year might be.
That's wonderful. Yeah, very exciting. I mean, everybody seems to be in a race here, but definitely I am biased for MGA to get to the clinical trial.
I used to hate xenotransplant.
I know you did. I mean, when I was working,
I tried to stay away from xenotransplant, but after starting that, I realized that, it's like just a regular transplant.
Yeah.
If it was.
But I think what has really contributed to the success of xenotransplantation is those genetic modifications that before we couldn't do, that can definitely dampen the innate immune responses that was not possible to do so.
This is great, both Seda and Dr. too. So quick question though, do you guys feel like you have enough information now about the pig models or the genetic modified pigs that you have, that once you kind of go through those last hurdles about the antibody treatment that Dr. So mentioned and something else, that you get the technique down and you feel that this is the last hurdle, or more issues are going to come up that we're not aware of? What do you guys feel is your knowledge of what's happening at the cellular level on these organs?
I think we have definitely gained a lot of knowledge from all these transplants that are being done. So I think we're exhausting all the other genes that might come up as deleterious that we need to knock out or knock in, especially from the pig side, the sugars. But really optimizing the function would be controlling, after controlling the innate immune responses, which Tatsuo mentioned, then is the co-stimulatory blockade. I mean, I should have also presented the paper that Tatsuo just published in AJT as well, with this data from eGenesis animals, that the co-stimulatory blockade, and especially seems like the CD40 ligand, not the CD40, so the other side of the receptor ligand interaction, seems to be more beneficial for xenografts to function properly, versus the heart needs definitely the CD40. So we still don't understand exactly what favors one pathway over the other for each of these xenografts. But at least right now, overall, we have the understanding that what we need for immunosuppression in kidney versus what we need for the heart, because the CD40 ligand is not sufficient to control a xenograft heart rejection, for example. They need the CD40. But again, tailoring each of those, so like I mentioned, CD47 might not be necessary to be expressed in the kidney because it might make it more prone to swelling and more inflammatory. So we don't have to have CD47 in there. But all the other genes that are important for the complement and anticoagulation, those definitely have to stay there. So those definitely are important. But that's what Tatsuo is trying to dissect out. How do you have consistency in each of the xenotransplant that he's performing for each of the 4 or the 5 of the 6 genetic modification animals that he's receiving from eGenesis. So I think that's why I said that they're pretty close to that. And that's why they might go ahead and apply for an IND approval with FDA so they can possibly try next year for a phase 1 clinical trial.
Said I'd like to thank you for an absolutely masterful presentation. And when you take a complex topic such as this and make it simple and reduce it to the basic elements, you've really succeeded in educating all of us as to the current state of this. Tatsuo, thank you for coming back on and also being part of this presentation, which I think really helped all of us get a better understanding of what you're doing and what Aseta has been doing for the last 10 years. I've always predicted that she has a phenomenal career ahead of her. And now she's got to get into medical school, finish that, and do the rest of the stuff.
No, I changed my mind. I just joined venture capital. So I'll be working on creating new biotechnology companies.
Now she got smart. Now she's gotten smart. Very good. Yeah. Now I've met a smart Albanian. Finally, it took years. But
I think all of this training is enough. Yeah.
She'll be great at whatever she does. That she's that type of person. And I wish her greatness in all of her career. And thank you for pushing the envelope, Tatsuo. Thank you for pushing the envelope that someday may allow people to clone me, hopefully before I finish. Nora, thank you for a great session.
Thank you.
Yeah, so I want to say that if you guys have any questions, I'm sorry if I could not answer everything from what was put in the chat. If you have any questions for me and Tatsuo, please feel free to email them and then we can just address them separately.
Aseta, thank you so much, Dr. Tatsuo. It's great. It's been an honor, pleasure, and so informative. I'm still like digesting, taking it in. It's amazing. It's great. And yeah, I might have a few questions. It's more like an ethical questions more than anything, since I'm an internal medicine doctor. But yeah, that was a great presentation. Thank you for joining us.
Yeah, thank you so much to everyone and for all your great questions. I really appreciate it. So
thank you guys.
Appreciate you guys taking the time. Thank you.
Thank you.
Lawrence.
Have a great day.
Nora, send me your emails, so I can send you your address, so I can send you those things.
I'm double thinking about that.
No, you need them. You're president.
Okay.
You know how I feel about the past.
No. It's Albanian Medical Society stuff. It's nothing horrible.
No politics. I
didn't. It's a packet they gave me. I'm giving it to you. I found it in my drawer.
Oh,
I haven't read it. I haven't even looked at it.
can Jürgen take it?
Pass it on to Jürgen.
I'll find it. Jürgen, I'll send it to you. Jürgen, give me your address. I'll send it to you. You can read it, burn it.
Oh boy.
If it must, then I guess so.
Do we have a journal club about that?
No. But you want me to throw it away? I'll throw it away.
I'll send you the mail. It's fine.
I'm happy to throw it away.
I don't have to open it. It's fine. I can pass it soon, very soon.
Just look at it.
Just
Come
pass it
on.
on to the next person.
Pass it on. Pass it on to Lawrence or somebody else and say, you know, that idiot coach, I gave it to me and I'm giving it to you.
Sounds good.
Excellent.
Thank you so much. This is
All
great.
right. Thank you, Dr. Tatsuo and Aseta, for a great presentation.
Thank you, Tatsuo. Thank you very much.
Hopefully we can have you
Like in
again
Boston.
for an update on that clinical trial.
Oh, yes. I think I will definitely personally invite Tatsuo when he initiates the clinical trials.
Yes.
Yeah.
Looking forward to that. This is exciting news.
Yeah.
Thank you, guys.
And Aseta, when you decide to go to venture capitalists, please give me a call.
She wants to invest in your company.
No, I'm not. I'm doing venture creation, which means that I'm actually building biotech companies for the certain indications. So I already chosen what I want to work on, which is on women's health. So addressing the unmet need there for many of the disorders of the reproductive system in women. So I'm building new drugs that actually are lacking. So that's my goal. You get new biotech companies and then new drugs that will cure the 190 million women that suffer from many of these disorders.
As you know, my mother died of ovarian cancer. You're too late to help our mother,
No,
but
I'm
help
sorry.
somebody else's mother.
I am hoping, to help
right.
those who actually will be affected in the future.
Good.
Very good. All right.
All right. Thank
Bye,
you.
everyone.
Bye-bye. Thank you, Tatsuo. Have a good day. Bye-bye.
Yeah,
Bye, Ted.
good job.
We should make Tatsuo an honorary member just for being here.
Actually, I also wanted to invite Dr. Kosumi to be here just because of you. But
Oh,
next
that
time,
would've been great.
next time I will.
Okay, I would enjoy that very much.
Yes, I think because his discussions and his opinions are very frank, so it definitely would have
Yeah,
been.
he's an honest gentleman.
I agree. But next time I will bring him.
Are you drinking vodka or water?
Water.
Okay. I'm just kind of checking up on my president. All
Early,
right.
early.
Both work. It's okay.
Bye, everybody. Bye-bye.
Bye-bye.
Bye, guys.
Dr.